Hello everybody, today we're going to look at bacterial cell reproduction. Bacteria are prokaryotic organisms that reproduce asexually. Bacteria can also reproduce sexually, and the sexual reproduction of bacteria occurs via a process known as conjugation. So sexual reproduction involves a sex pilus, which is actually a mating bridge that is formed between two bacterial cells. With the aid of the sex pilus, they actually exchange genetic material. No new cells are actually produced, but their genetic makeup essentially changes. But today we're going to focus on asexual reproduction. Bacterial reproduction occurs most commonly through a process of cellular division known as binary fission. So bacterial reproduction asexually. So most bacteria reproduce asexually by binary fission. So during binary fission, the single DNA molecule actually replicates. So both copies of the DNA then move to opposite poles of the cell, and the plasma membrane then begins to grow between the two DNA molecules. So once the bacteria is double its original size, the plasma membrane begins pinching inwards, and a cell wall then forms between the two DNA molecules that divide the original cell into two identical daughter cells. Bacterial growth is normally affected by factors such as temperature, pH, food and nutrient content, etc. I'm going to look at temperature and pH. Temperature classification based on temperature. Psychrophiles are cold-loving bacteria. Their optimum growth temperature is between -5 degrees Celsius to 15 degrees Celsius. Mesophiles, on the other hand, grow best at moderate temperatures. Their optimum growth temperature is between 25 degrees Celsius to 45 degrees Celsius. Thermophiles, on the other hand, are heat-loving bacteria. The optimum growth temperature is between 45 degrees Celsius to 70 degrees Celsius. pH classification based on pH. Acidophiles, which means acid-loving bacteria, grows best in an acidic environment, usually at pH less than 5.5. Neutrophiles grow best in a neutral environment at a pH range of 5 to 8. Alkaliphiles are alkali-loving bacteria which grows best in an alkaline environment, pH more than 8.5. So this is bacterial growth, and you can see it is divided into 4 phases: the lag phase, the log or exponential phase, stationary phase, and the death or decline phase. So during the lag phase, growth is usually slow as the bacteria acclimatizes to the food and its new habitat. And then during the log or exponential phase, once the bacteria has managed to acclimatize to its new environment, its growth is exponential. Next, the stationary phase, and during the stationary phase, the supply of food and nutrients begin to dwindle. The booming growth comes to a stop, and the number of bacteria stabilizes. And finally, the decline phase. During the death phase, the bacteria begin to die. This is because of toxic waste accumulation or buildup, as well as its food and nutrient resources has been depleted. With that, I thank you.